Imagine a bag filled with 100 marbles, half red, half blue. Now, randomly pick out 10 marbles to start a new bag. Maybe you end up with 7 red and only 3 blue. Just like that, the balance has changed. Not because one color was better, but just by random chance. This is the idea behind genetic drift. Unlike natural selection, which favors traits that help survival, genetic drift happens purely by chance. Genetic drift is the random change in the frequency of alleles in a population. But what does this mean? First, let's understand the key terms. Alleles means different versions of the same gene. For example, one gene might control flower color, with one allele for red and another for white. And frequency means how common each allele is in a population. For example, 60% red allele and 40% white allele. Now, the full meaning. In every generation, some individuals may reproduce more than others purely by chance. Not because they have better genes, but just due to luck. As a result, some alleles may become more common, while others become rarer or even disappear, not due to their usefulness, but just by accident. So, when we say genetic drift is the random change in the frequency of alleles in a population, we mean sometimes, by pure luck, certain versions of a gene become more or less common in a group, not because they're better or worse, but just because of random events in who reproduces. There are two special cases of genetic drift: the bottleneck effect and the founder effect. The bottleneck effect happens when a large population suddenly gets much smaller because of a random event, like a natural disaster, disease, or hunting. Only a small number of individuals survive. These survivors are not necessarily the strongest or the best adapted. They were just lucky. Because of this, the new population may have very different gene frequencies compared to the original group and much less genetic diversity. The founder effect happens when a few individuals leave their original population and start a new one somewhere else, like on an island or a distant area. Since only a few individuals are involved, they carry only a small part of the original population's genetic diversity. As a result, the new population may have different traits than the original one, simply because of the random genes the founders happened to carry. Let's look at two real-life examples where genetic drift shaped entire populations. First example is cheetahs. Cheetahs have experienced a severe population bottleneck. Thousands of years ago, cheetahs went through a massive population crash, possibly due to a climate event or disease. Only a few cheetahs survived. Today, cheetahs are so genetically similar to each other that scientists say it's almost like they are identical twins. This lack of genetic diversity makes them more vulnerable to diseases and environmental changes. Second example is Amish people. In the 1700s, a small group of Amish people migrated from Europe to America. Because their founding group was so small, certain rare genetic conditions, like Ellis Van Creveld syndrome, which is a form of dwarfism, became more common among them than in the general population. This wasn't natural selection. This was simply the random genes that the founders happened to carry. In both cases, random chance changed the course of a population's genetic future.